One. Hello!
Hi there.
Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny. In a previous life, I dropped out from my PhD in microbiology. Then I was a fact checker and editor for pharmaceutical advertisers. And nowadays, I'm a member and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to aim at the public with the tools of biotech.
My name is Faz, and I have a PhD in microbiology, where I mostly worked on bacteria. Since then, I've worked as a research integrity specialist, and I'm currently working as an editor for a scientific journal.
Every other week, we meet to talk about microbiology, and on a typical day like today, we're doing an overview of some of the coolest microbiology papers that we've seen in the last 4 weeks this time, since we had a little
that's right.
hiatus.
Also, we want to do deep dives into viewer-selected papers, so leave us a comment, toot, or tweet with your suggestions for a deep dive, and we'll look into making that content.
Yep, you can follow along with the papers that we discuss in either week in our shared Zotero library, our social media handles, and the link to the library in the doobly-doo below.
And boy, do we have a show for you today. First, we have our SARS-CoV-2 stories, where we ask, how does SARS-CoV-2 enter the brain cells, and what does it do when it gets there? We also delve into the war between bacteria and phages to see how they evolve new weapons. And then we look at the discovery of new multicellular bacteria from an underground cave. And also we see how researchers can attach backpacks to bacteria and use them to treat diseases. But wait, there's more. We have researchers who have figured out how to take pictures of viruses at the earliest stages of infection, and we also get to look at those results. Also, we see how antibiotics can crystallize bacterial cell walls, why phages make their own tRNA, how integrons can transfer genes from Archaea to bacteria, and so much more. So please stay tuned. But first, we're going to be looking at our SARS-CoV-2 stories.
Yep, the first one we have is called Lifting Universal Masking in Schools, COVID-19 Incidents Among Students and Staff.
Yeah, so part of the reason why we're off for longer than usual is because I caught COVID because I got complacent. Yeah, I got complacent. I went to a movie. What was it? Yeah, a movie theater, and I took my mask off. And usually, I've done this before, but the problem is there was I discovered this only about 15 minutes. But there was an old lady near us who was just so ill. It was very loud, ill that it made me put my mask on halfway through the theater. I put my mask back on in the cinema. It was so disgusting. And then
that's a tough story because you could kind of see it already was happening, unfolding in front of you. You think that was the event.
I'd been hit by that car, and I was just like, okay, so yeah, it happens sometimes. I think that so I got complacent and I didn't put my mask on, and so that's what kind of got me picking this paper, because this paper is looking at universal masking in schools, because it looks into what we call a natural experiment, where towards the end of I think towards the end of the outbreak, schools were naturally lifting up the mask mandates. So in Massachusetts, they removed the legal mandate to wear masks during in school, and some schools took it off immediately. Some schools waited a while, and so this paper looks into comparing what happened in each of those schools. How long did it take for a COVID outbreak to come up in schools after they after they lifted the universal mask mandates.
Yeah. So the natural experiment refers to this idea that it wasn't like the experimenters created these two conditions and we're trying to compare them. It was that this is something that's already happening in public policy. And if there are differences in the way that the schools, I think it's school districts that they're looking at here in Boston, if there is different ways in which those districts are approaching the policy, then maybe those differences in the way they approach it could show us differences in the actual concept of having masks on in schools. And so this was done in 2022, which I think I just wanted to say this year I think has been sort of a lot more people have gotten COVID just because there has been this attempt to return back to a less stringent condition. And in doing so, it's kind of just thrown a bunch of chaos. Plus, because there have been new variants, right? The BA4 and BA5, most notably, right, drove a large increase in cases this year. I think we kind of got to see what does it look like when there are fewer non-pharmaceutical interventions, and there is sort of an influx of cases. So yeah, so basically they only have like a 3-week, they only have a 3-week difference between most of the schools, right? They have 2 districts that never remove the masks, the mask mandate.
Yeah.
And then they have like a cohort that removes it immediately in week 1, a cohort that waits 1 more week, and then a cohort that waits another week after that. So that's the Q. Oh wait, is it Q? I can't see the letters.
Yeah. Let me. So like the.
Oh, so just, like it's just 1, 2, and 3.
Yeah, one, 2, and 3. I think are the times that the mandates were lifted,
yeah. And then the different line colors just correlate to either the dark gray line is the sustained masking. That's the 2 districts that stayed for the whole time. Yeah. Then the lighter the color, the earlier they do. And actually the end numbers change quite a bit too. Like the earliest ones, there's 46 different districts that unmask in the first week.
Yeah, and I think there are also some like confounding factors that we need, because I think usually when they will just the schools that kept the masks on usually did it for a reason, usually because they had like very crowded classrooms and bad ventilation as well. So there was always this sense, the idea that maybe those effects would also overshadow the masking. Perhaps they did some very fancy statistics to kind of see kind of to control for when the mask mandates were happening, to kind of like normalize them. So moving each of the data points so that like as if it's one they all like lifted at the same time, and found that generally that there was a spike at a specific amount of time after the mask mandate was lifted.
And yeah, and I mean actually one of those differences that you're saying, like more crowded schools or like worse ventilation, those schools kept their mask mandate, right? Like, or so actually, if we were to see an effect in them, right? Like you'd be like, oh, like that means that like those crowded classrooms and that poor ventilation was like really affecting the numbers, but actually they have the least spike, I think.
Yeah, that's right.
Yeah.
So essentially what this kind of shows is that the mask mandates tended to do their job, as in for that the moment you lift them, you're going to get a coronavirus outbreak, and the earlier you lifted it, the kind of the worse that outbreak was.
Yeah. So it actually makes me think, like, at least in the States, people are talking about like a lot of respiratory viruses are going up right now. Like it kind of all happened at once. And I wonder whether or not it has like a similar idea that like the removal, like sort of like a collective action to take off masks, like just meant that now everything's sort of back to normal, and that's, or not normal in the sense of like viruses can easily spread between us again. And so, they're just having like a heyday.
Yeah, I mean, we've got a very nasty flu like outbreaks happening. I mean, there's avian flu going on around as well, which I guess isn't really linked to masking at all, but is going to affect people over like Thanksgiving's over, so that line doesn't work for the script now. But for Christmas dinners and stuff, there's going to be some problems with like shortage of chickens and other things.
Yeah. Oh, I see. Interesting. Yeah, I wasn't thinking about that. But anyways. Stuff sort of applies, or like the takeaways that people can take from this are that, you know, masks work, which I don't think we never questioned on this channel, but like, if you were still doubting, like, here's some more evidence to say that it is a useful intervention. The authors, I think, in the discussion have like a very interesting point. They actually try to specifically talk to potential criticisms, right? They say, oh, but what about people who say that the masks are disruptive for like school classes and so forth? And then the authors try to point to data. They say, well, all these people that got sick, they got disrupted classes. So then really, like, what's the logic in that? And then they did a second common criticism. They said, oh yeah, it was the criticism about these like better ventilation and right, like better ventilation and smaller class sizes, those things can help prevent what was going on. But as we just said, that the two districts that kept their masks on for the entire time have the worst ventilation, the highest class sizes, and they had the least effect as it was studied here.
Yeah, so I think that's like, I just want to sum up that, like, kind of, we've always like a cow goes moo, a sheep goes bar, a Phage goes wear your mask. So it's like, Except when I forget that as well, because we're all human. Sometimes like that stuff happens.
Absolutely. And I think at the end of the day too, like it's sort of like the natural experiment aspect of this I really enjoyed, because we're not in full control over all of these policies and sort of things. I mean, through activism, we can try to like raise our voices and so forth. But, you make a scientist looking at this sort of problem tries to make the best of it and says, well, we could maybe potentially at least get some data out of this scenario in order to inform the future decisions, right? Maybe there's future decisions that will be impacted.
Yeah. Anyway,
okay.
Next paper. Neuropilin. Sorry, I'll let you do this.
Go ahead. Okay. Neuropilin-1 mediates SARS-CoV-2 infection of astrocytes in brain organoids, inducing inflammation, leading to dysfunction and death of neurons. So we've talked about SARS-CoV-2 getting into neural tissue before. Of course, people are very concerned about this because of the post-acute sequelae. I think they call that PASS of SARS-CoV-2 or long COVID, so forth. Also, like when we identified like the loss of smell and things, that seemed to be linked to some sort of neurological stuff. And then this takes something that I really enjoy, organoids, which is growing cells in a cell culture that recapitulates some of the complexity of tissues. So organoids are really cool. Brain organoids, they're actually inhabit this kind of creepy space where ethicists have had to come to terms with like, well, what if it looks like a brain and it acts like a brain, then what if we're making tiny brains? They use human-induced pluripotent stem cells. There's actually like a research ethics moratorium over like letting these organoids develop to like a certain stage, because it's kind of like a gray area. But anyways, these ones are ethically sourced, presumably, right? Like kind of before they get too developed, and they have all sorts of tissue in them and all sorts of cell types, which is also very interesting. So astrocytes are these like supporting cells that sort of nourish the neurons, because neurons don't have a lot of their own metabolic pathways active inside of their cells. So yeah, they're like sort of surrounded by a bunch of other useful cells. And they infect them with SARS-CoV-2. And I guess the big part about this, and it's linked back to something we've talked about before, is that not all cells have lots of ACE2. And especially like in the brain, there's actually not that much ACE2 contribution on astrocytes or neurons. But they do have neuropilin-1, and neuropilin-1 is a receptor. We saw that. There's like a back-to-back paper that we covered at some point saying this.
Yeah, that's right.
So they infect these cells, and they look at a bunch of gene expression changes. They look at what cells get infected, what genes are changing, and in general, it's what you would expect from an infection. Let's see. I think they say expression changes induced. They have interferon pathways get induced. Inflammation pathways get induced, and then things get downregulated, like cell-cell communication and cell junction organization. And what that ends up looking like inside of these brain organoids is cells die. Cells die in the organoids, and the organoids also kind of fall apart a little bit. I think if we go to the final figure, they have the most dramatic figure 6.
Figure 6, let's go.
Figure 6. Yeah, in figure 6, they do some like apoptosis, looking for signs of apoptosis. And so those are coming up in green. The Tunel assay looks at like a specific pathway that gets activated to chop up cells on their way out. And in the SARS-CoV-2 infected organoids, it lights up. And I don't know, this one in panel C, I think it kind of made me laugh because I don't really know what it's a funny, it almost seems so dramatic, right? To be like these little organoids, they also start falling apart.
I think some I just want to like kind of, let me zoom in on this quickly. Let me see if I can wait. Really get this point.
And just
there is a very much a sensation of this is your brain, and this is your brain on SARS-CoV-2.
It is, and just to remember, like that's not these are cell culture models, so like it's not exactly the same, but of course, like this is the implication that something like this, or at a small scale, like disruptions of this nature, are not good, and so
not gonna lie, now that I had SARS-CoV-2, I think that's a little bomb panel makes a lot of sense. Just I did that were not smart at all during when I was.
So yeah, again, stuff that we already know, but it takes a lot of time to set up these model systems and gather the data on them, and so it could be useful, I think, because they do look at in this paper some pharmaceutical interventions of some sort, trying to block neuropilin-1 in some ways. Like, I don't really know what that does in terms of the cell signaling side into your body, but there's some suggestion that the information they find might be useful in some therapeutic way.
Well, I think it's also like important to nail down that neuropilin-1 is an entry factor, and because it wasn't necessarily 100% certain when we first looked at it. And I think my interpretation of that was that they really wanted to nail down exactly how SARS-CoV-2 was entering and whether neuropilin, because I think neuropilin, you have this weird like C-end rule kind of peptide that SARS-CoV-2 somehow attaches to. And it seemed a bit like of a kind of a stretch, but now like they've got all this evidence that is stacking up about neuropilin being so important for that.
Definitely. And I think we had covered some papers before about SARS-CoV-2 in the brain, and they were also talking about like leaky endothelial stuff, interactions with the endothelium and astrocytes, and so this also fleshes in that story a bit more, because we're now talking about other activations, other inflammation activations that could be happening. And so, like, again, the experts that are studying this in their field, like, this is like really useful information to build more detailed hypotheses. But for us, I'm just kind of like, oh yeah, more brain stuff. It's like solidifying that in my head.
And I think there was another like protein that like kind of they mentioned also was like helping, like the endocyte, one that was related to endocytosis that kind of pinged as well. And they also compared with the different where there were some strains that are more damaging to the brain than others. Which was
that's right. And they saw that the Omicron, and we heard this before too, right? The Omicron didn't cause as much issues as the other strains.
So I mean, goodish news, I guess. But
it's hard to say, right? Like, I keep thinking back to, throughout the course of this whole pandemic, I keep thinking back to, like, we looked at this paper that says, like, you don't know whether the next variation is going to be worse or better, right? There wasn't a good predictive capability there. And so, I mean, in this case, like, we really only have the vaccines to rely on. So, like, making sure, like, you're updated with your boosters and stuff gives you, like, some protection there. This is just random, but like, I got the Novavax booster. That was like an option to explore. And I was like, okay, I'll give that a shot.
Yeah, no, it's been a year since I got my last booster, so I think that. So
but now
probably should have gotten one.
But now you had natural infection, so.
Now I've got natural immunity, which is just great. It's so much fun. So really, really enjoyed that.
Okay, let's move on.
I'm not bitter.
All right, let's see. Next thing we got.
The
evolution of counter-defense mechanism in a virus constrains its host range. So we're not talking about SARS-CoV-2 anymore. Now we're talking about a phage system. So the virus that they're looking at is T4 phage, or part of the broader family of the T-evens, if you will. So T4, T2, T6, and T8.
What a neat way to classify them.
I know,
it's like they're rating the Star Trek movies.
Also, Minref added in the comments if anyone saw, the astrocytes are also weird. Yeah, astrocytes are like a really weird cell type. There's support cells. They don't have to enter the cell surface to infect them. Yeah, I'm curious about that. If you want to type in the comments a little bit more information about there, I would assume that might mean like the syncytia thing. I don't think they looked at that in the last paper, But that is sort of a huge part, right, about Omicron that we had talked about, that particular phenotype of forming cell syncytia means that they could affect neighboring cells without necessarily having to have a particle attached to the surface of it.
Yeah.
Okay, sorry. I just saw that now and it distracted me. So yeah, so the T evens, trying to just figure out like bacteria have defense mechanisms against phage. So of course, phage probably have ways of defending against those defense mechanisms. This is like the story of pathogenesis, sort of an ongoing arms race, if you will, coevolution, if we're being more holistic feeling, I think. I like that language a lot better. And so they essentially go in and they have wells of bacteria that doesn't have a particular antiphage system called tox-IN. And they have cells that they do have this defense system. And they serially passage their T4 phage inside of it until they start seeing the emergence of some phage that are able to grow in the bacteria that has that phage defense system. Yeah, and what they find. Oh,
sorry, no,
yeah,
go.
Sorry, you want to? Okay. What they find is that they find two genes that are particularly impacted here, called DMD and TIF, or well, they don't know what that is. They later call it TIF-A, but they just call it like, open reading frame 61.4. And so they find these two things. DMD they know is potentially an antiphage system, or sorry, what are they calling these? Like a overcome media defense, anti-defense system, virulence factor. I don't know.
A counter defense system, I think. That
counter. I like. Yes, that's a good word. Counter defense system. Yeah. So they know about this DMD counter defense system. They don't know about this 61.4. Sorry. Go ahead.
Yeah. I mean, also like because as I understand it, they found this entirely new like defense system that kind of like cropped up through their manual evolution, essentially. And I think they be like one of the like kind of genes that didn't have much of a function, and then there's a recombination event, and that gave it a function.
Well, I think what it did, what they found, they didn't have they didn't have a function for it beforehand, but in studying it more, they realized, okay, so what were the mutations that happened that let this particular gene somehow work better? And they were finding duplications, like a type of there's like multiple copies. It's like under a different promoter. It's like looking at it, it's being accessed by another promoter. So like there's more. The idea is probably that there's more mRNA of this particular thing, and that's giving us more protein. Because they do look at it associates, so the protein associates with the antitoxin or the toxin antitoxin system, and so like it is an interactor, and the idea is like in cells that are very resistant, they have tons of this particular toxin antitoxin system, basically saying that upon phage infection they take one for the team and die. But these phage, they encode multiple copies of this TIFA or the 61.4, and that makes enough of this protein that sort of sequesters that suicide signal or that toxin antitoxin system.
Okay.
Yeah, so it's about I think it's about the duplications and having more of them. They do, I think, look at a case where so it is duplication. So there's more genetic information there. And so relating back to the title of this whole paper is that they say, so like how do you even get a duplication? And what they see is that some genes get deleted because you have to make space for these extra copies of things. And the genes that end up getting deleted are typically ones that are used in other phage defense systems. So when they look at some of their evolved clones, they then use like different bacteria. I'm trying to see, where's the best figure to show this one? Oh, I think you might be on it already.
Oh.
Yeah, no, not figure 3. Figure 4?
Figure 4, let's go on figure 4.
Figure 4. No, figure 4 is just a bunch of clones. Might be later on. Let's see.
So
there's a lot of figures in this paper. It's a big one.
Yeah.
Oh no, you were on the right one. Figure 6. I'm so sorry.
6. That's all right.
Sorry.
You got a nice little tour with the other figures.
Yeah, like in figure 6, this is where they say, okay, well, like they lose other genes, and that makes them less likely to grow in some of these other strains. So they have these other strains of bacteria, ECO17 and the wild type MG165. So essentially, like they've swapped out the ability. They became extra in the course of this experimental evolution. They became extra specialized to be able to overcome one particular toxin antitoxin system, but in doing so, they lose the ability to grow in some other host cells. And so this is an evolutionary biology paper, right? So they're trying to say, like, this is maybe a trade-off. This is the fact that recombination is happening in these cases. These are like some of the mechanisms that we could expect viruses to do in order to find new host ranges to explore. And this could also help us explain like some of the genes that we don't know anything about. Like maybe those are very specific for specific hosts, right? It gives us this kind of like thought process.
Right. Yeah. Okay. Yeah, that's quite cool. I mean, almost like, yeah, it's always interesting to see these arms race papers. I know you prefer coevolution. I prefer arms race. I mean, potato. Let's kick the whole thing off. But I mean...
yeah, I mean, it's so complicated, right? Like, there's so many interactions that are happening in this microscopic world, and those genes are probably tools in those interactions, but without seeing all of those interactions, yeah, we kind of don't always know. So it's nice to see when they can piece apart a little bit of the story.
Yeah, and it'll be quite interesting once, because based on those toxin genes, they can try and like estimate what bacteria that, because I can imagine like some metagenomics coming out of this kind of research where they go like, oh, we found this phage, so then we can assume that there are these bacteria that it also infects maybe as well. Or
yeah. Kind of... The authors talk about how phage genomes are super diverse. Like there's like a huge accessory genome essentially to all phages.
Yeah.
And again, this is like part of the explanation. Like all those accessory genes are because like these phages are having to deal with so many different host defense systems that they have to swap in what they need. Like they have to swap in a huge diversity in order to be able to infect a diversity of things.
Yeah, and I think it also helps to have that diversity there in case they encounter something completely new, because it gives them that kind of depth of like, it gives them lots of options in how to deal with different host antitoxins. If they've got this entire library of other things they can dip back into and recombine, and it gives them a lot more to work with if they're dealing with something completely new.
Totally. Okay, so moving on, we got. Novel multicellular prokaryote discovered next to an underground stream. Yeah, this is a fun paper. I think that it's very much just like found something in a cave and grew it in the lab, and it looks weird.
Yeah, I mean, even like finding something in the cave, because this is like an underground river. So I don't know, like I don't know if anyone else has a worse fear, but I feel like up there is like cave diving.
Definitely.
So. Yeah, this was like isolated in 2008 in like the Blue Dragon Cave in Japan, where they sampled some site of like this limestone wall that was close to the surface of an underground river. So like water will go up and down. So sometimes it'd be submerged, sometimes it wouldn't be submerged. And these scientists imagine
scroll us down to that figure
that way just for that one sample, or I don't know what.
Figure two.
Oh, figure two.
Oh, it's an image for the backdrop of the story you're telling.
Yeah. Yeah, like a cave drowning in complete darkness. I mean, a caving in. Yeah, just.
Yeah, but these guys, I guess, have the wherewithal to collect samples. I mean, when else are you going to be in this scenario?
I mean, they probably assume they know what they're doing. They're not like me and panicking and hyperventilating, but. But yeah.
So they pick up a bacteria, and when they grow it in the lab, it has this in panel B. You can see it has this like crazy iridescent color profile going on, and so that's really weird. And I guess, well, they were going to do the investigation anyways, whether or not it was iridescent. And so they look at microscopy, and it has like some different morphologies. There's cells that are long and sort of ordered, and then they have bulges in them. The cells have a flagella, and yeah, so it's like they sort of do a full description of these cells, and they're most interested in, well, why does it become iridescent, and why does it have these different like body form types? And I think a little bit later they figure out like the iridescence is caused by oh my God, what's this word? I had to look it up.
I think they mentioned pneumatic liquid crystal.
Pneumatic liquid crystal, yeah, and anisotropic.
Yeah.
Anisotropic optical structure. So what that refers to is just that different sections. You can see it most clearly in D, I think, where they use like some coloring to help us differentiate. Essentially, they're looking at the colors are based on the angle orientation of the bacteria. And so that they're saying like in these little clusters of bacteria, they're all angled in the same way. And then in another section, they're angled in another way. And it creates this kind of funny crystalline lattice. And then they're thinking that is what is responsible for the cool colors. Because yeah, it's just like letting light through in a specific way. Yeah. And yeah, I don't know. I guess like it's a peculiarity. They're saying that it means that there must be some sort of organization on a sort of colony level that's interacting in order for them to take on these specific shards of orientation.
Yeah.
And then
I mean
sorry. Go ahead. No, go ahead.
No, you talk on this because I think you've got a bit more of a background on it than I do.
Okay, I won't move to figure 4 then. Because then the other thing, it's just another thing. So another thing they describe in these colonies is that they find that in the middle of the colonies, after they let the colonies grow for a period of time, then they see more of these like the bulges, more of these cocci-looking cells. Instead of like bacillus, instead of long cells, they're becoming more like ball-shaped, and they're really like getting denser in the center. And when they rinse these colonies with a bit of water, the water specifically pops the middle of them and then lets those small cocci cells like flow out. And then if they take those cocci cells and they plate them again, they go back to the sort of weird crystalline structure before like they develop. So essentially, seeing these phases gets the authors to say that they are developing. It's like they're working together to create this specific structure so that they can propagate themselves, and that probably is related to the environment that they're in. The fact that this cave gets flooded periodically. It's like, I don't know, they got a sticky form and then they got the dispersal form.
Yeah, and they got some brilliant videos on here as well. Like, so they show the dispersal of how. So the idea, I guess, is that it's on the cave walls, it grows and grows, and then when it gets submerged, and instead of just growing bigger and bigger, it lets fly a bunch of like I don't want to use they're not spores, but bacteria to like almost act as seeds for a new colony, which is again quite a great demonstration of like how microbes tend to use multicellularity, because I think we're quite familiar with like fungi who like can form like really impressive like spore dispersal mechanisms and fruiting bodies.
Yeah. Some protists, the dictyostelium.
Yeah,
right? It was my favorite little model. Like they have that whole like slug, and like then they get starved and they make a stalk. But here we're looking at a prokaryote, so that's kind of cool. But like the whole paper is framed in this way, right? Saying that like multicellularity is something that scientists have come to understand has happened many times. Because it is just like a useful strategy in a lot of different scenarios. It's a type of like close cooperation, right, between cells growing together. So here's just another example, and who knows, like studying this may teach us more things, because I think at the back of people's brains, especially like the biotech field, it's like, well, how do you make these single cells do things that we want, right? And how do you make them like go and tackle certain problems, complex problems, in a way that is efficient and like uses less resources? So like imagining, like learning about these multicellular things, you could try to like maybe harness that for ourselves. And then again, in a not so human-centric way, it's just cool. Like diversity of life is amazing. Like I want to see more. I want to know how all the things on the planet work. and these types of stories are just really cool from that perspective.
Yeah, I mean, I like the whole exploration aspect of you going to somewhere strange no one's ever been to before, seeing a weird iridescent bacteria, studying it and finding out how that works. That's like the fun explorer part of microbiology.
Totally, totally. It's like out in the savannah. I know. I always think about this. Like, there's so much people like are like really interested in all these animals and their crazy lifestyle and their behavior, but it's like happening on the micro scale as well. It's just kind of harder to access sometimes.
Yeah, and I feel like there's so much diversity and like so many different stories on that microbial level that are waiting to be like found out. And this is kind of paper that gets me excited about that because I mean just looking at this bacteria on a plate is just it's like it's from an alien planet. I mean, if that was if this was in a sci-fi movie, this would be the thing that kills human civilization and turns into zombies. But because it's real life, it's bacterium.
Yeah. It's just doing its thing.
Yeah, it's just doing its own thing, just like hanging out and being weird.
All right, so let's move on.
Yeah. Next paper is Mucoadhesive probiotic backpacks with ROS nanoscavengers enhance the bacteriotherapy for inflammatory bowel diseases. So this is a paper that I mean the title itself, probiotic backpacks, is such a weird term because bacteria don't have back. Oh, I guess like there's a different pun in there that I missed. Bacteria packs. Oh, okay.
It actually.
But the idea is that in the intestine that affect the gut lining, and and trying to get drugs into that gut lining, it can be a bit challenging. But one things that do get into a gut lining very well are probiotics, like bacteria. They live there. So what if we got bacteria that naturally like to live in the gut and attach some cool compounds to it? In this case, they attached a reactive oxygen species scavenger that basically reactive oxygen species are a signaling mechanism during inflammation. So often when you get lots of inflammation, you get all these reactive oxygen species going around, and they can destroy bacteria, but also they can cause a lot of stress, and so they can be quite bad in too high doses.
Yeah, I mean that's the double-edged sword of inflammation always. That like inflammation is our immune system trying to deal with things, but it ends up like causing collateral damage in many cases, and depending on how sustained it is, it's just not good for our health.
Yeah, and so you got a disease like inflammatory bowel disease where you've got like the inflammation is just it's getting too much.
Yeah.
And so you want to calm it down. And so the idea here is to create these scavengers and deliver them to the bowel to calm everything down. And then so part of this paper is just looking at the chemicals of taking a known ROS scavenger and then turning it into almost like a polymer. Yeah. They get lots and lots of these things lined up to kind of grab as many of to neutralize as many of these reactive oxygen species as possible.
Yep. Yeah. So that's the backpack.
Yeah.
is the polymer with reactive oxygen species.
Yeah. So that's like kind of the cargo they want to deliver, and then you have to strap that to the bacteria by creating a special coating for the bacteria, and that coating itself is made out of norepinephrine, which I thought was like a neurotransmitter. Apparently, it's very sticky, and it's found in like the giant snails. Like kind of, there are certain snails that use it to stick to things. So they tagged like norepinephrine with this PPS, which is the antioxidant, and then added it to this is a hyaluronic acid. And coated bacteria in them and sent them into the guts of rats to.
Yeah, and we've seen this. We've seen a lot of these things before in some ways. Like the E. coli the E. coli that they're using is Nissel, which is like one of these like model probiotics. I think I'm remembering I picked out a paper where they use it in chickens.
Yeah, that's right.
Right. They put it inside of the fruit caviar. This is when you didn't know about fruit
caviar.
Yeah,
the fruit caviar. I'm learning. Yeah.
And then they put these bacteria in it, and so so this is like a probiotic. And I think another part of giving these bacteria, and then they coat the bacteria, which is actually something we saw earlier. They did a coating to protect the bacteria from antibiotics. Do you remember that paper?
Yeah, I remember that paper.
And so that's kind of hilarious too, because like here they're like doing a coating technique in order to like essentially protect these E. coli, because the E. coli could be damaged by the reactive oxygen species as well. So it's as much as like dampening down our inflammation as it is also like letting these bacteria have like the tools so they can damp down inflammation and then enter these crypts that are and colonize them. Yeah, so it's very much like suiting up your probiotics in order to make a difference in inflammation. And they use the DSS model of colitis here.
Yeah.
So that's a
So yeah, they feed them like dextrum salt. I can't remember what DSS stands for, but it's bad because if you eat it, it gives you colitis. That's all you really need to know about DSS. I think,
and scientists, I think scientists love it because it's like you dissolve it in the water at your favorite concentration and you feed it to mice, like for a very specific term, and it creates like very reproducible like colitis symptoms and morphology. So then when you have something you think is gonna help with that, you have a great disease model, essentially, a very simple disease model.
Yeah, and I think they basically find out that when you give it to rats, it helps them survive the deep colitis better, and like kind of they don't lose as much weight, and that's I think the main
you're saying rats a lot. It was rats? I thought it was
no,
mice.
so it was mice, but I get those two things confused because I'm gonna use COVID as my excuse here. Okay, I had okay, man, get off my back.
Yeah. So it works. The backpack works. It helps the E. coli get in. I think it makes their guts longer, which is like sort of a hallmark. Shorter guts is like a hallmark of having a bad time with colitis. It works on a lot of different axes.
All right, so next paper is, this is an exciting one. So exciting that I'm going to have to switch, because this paper is titled Capturing the Start Point of the Virus Cell Interaction with High-Speed 3D Single Virus Tracking.
Which actually from the title, I wasn't entirely sure if I was going to super enjoy it, but in reading it, I was just seeing, they're deploying some pretty complicated technologies. This is those instances in science where you're like, wow, scientists can be really precise sometimes with their tooling. Like, I mean, it's not CERN level of like sensitivity or whatever, but like I think it's getting pretty close. Go ahead, Fuzz, please.
So I mean, let me like highlight the problem. So what these researchers want to do is they want to capture viruses just like as they're hitting the surface of the cell, like the real start of infection. The problem is that trying to seeing things with that magnification is really difficult, because either like if you want to get the long view, it's like imagine you're trying to film a racetrack, but you don't have the ability to filming like an F1 race, and you don't have the ability to move your camera. So you have the option of zooming out so far that you can barely see any of the race cars, or you can zoom in really fast and you catch a race car for like a fifth of a second as it rolls past the camera. That's essentially the kind of problem that people have when trying to photograph viruses as they're going around, because diffusion is deceptively slow. Because something can take sometimes to diffuse around across the room, but the virus in that time is actually just bouncing off of all these other molecules. And it's so what these researchers have kind of devised the ability to do is to follow viruses with their camera. So they use some really fancy technology I'm going to show here, because what they have to do is they have to move the entire microscope to follow the virus. And so let's say you've got like the researchers produced this amazing video. So you've got this like sample holder and the stage, and you see that there's a virus over there. And they have to and this like the cyan bit is the microscope trying to find the virus. And once it like kind of finds the virus, it needs to be able to find its position and track where it's going. So that's why it has this kind of, it zooms around a lot, and when it like passes the virus, it maps like the area where it's going, I think, as well, and essentially moves the microscope slide to track it. So it creates almost like this volume.
Yeah, it combines that, right? So like that's the one way in which it gets like to find that one particle. Then it has to like contextualize that within the whole space that particle is moving in. So they're essentially using two microscopes here. One that's going to take the zoomed out view, and then one that's doing this like kind of tracking of that tiny particle.
Yeah, and the zoomed out view camera has to be very, very fast, because for confocal microscopy, you have to take things at multiple. You have to take multiple slices of image. So that means you have to essentially like be moving your zoom lens like in all sorts of different directions. And usually in microscopy, or at least in the microscopes I grew up using, you have to like switch entirely new lenses every time. Whereas they got these new fancy electrically controlled lenses where they've got these liquid lenses that they run an electric charge through that can change their shape. Allows them to create to image like the entire volume of like a cell very, very quickly. Yeah. So the first thing, the way they track the viruses.
I was gonna the entire volume. I guess it is the entire volume of the cell. No, it's kind of constrained. Something.
So they use to track the virus, they use this thing called active feedback tracking, that's how they track the virus. And then in order to get an idea of what's happening else, so you've got these electrically tunable lenses that also get like an image of like the area around that virus at the time, and then from that they can build up an image of the cell that they're looking at.
And so I think one of the first things they do is they compare like, what if they looked using a regular confocal, spinning disk confocal microscopy as like the baseline way that people do microscopy, and they're like, well, how does that compare to what we're doing? And they get a good they see that like when they use spinning disks, like you can't see these, like diffusion is like it's wiggling around, and so like when they use spinning disks, like you just don't get that type of resolution. You get like more jumps, and so like it just looks like it's like lines, I guess, less of a wiggle.
So the great thing about when you get new technology like this, firstly, there's understand, and then they just point it at cool stuff and figure out like what's going on.
Sure. Yeah.
So I mean, here
this is one of the wiggles.
Yeah, this is the wiggle. So this is kind of how you see like so. So at the moment it's like freely diffusing. So diffusion, it's bumping off of shit. It's running around very fast, but it's zigzagging in different directions, and then it bounces off the surface. So that's what is called like skim. So the idea is that the viruses don't immediately latch on and go in. They kind of like skim the surface for a bit and like bounce off.
It has to find the right
receptor. So kind of those yellow patches.
Receptor, right? Like to increase its interaction probably. Yeah. So if there's nothing there and it hits, then it just bounces off and it continues on its thing. But it's like, if it does end up having a receptor, all of those skim events, someday it'll have an event where it lingers a bit more. Oh my gosh.
It could even be attached to the receptor, but it's just like it has too much energy and bounces off immediately, or kind of are going on here. And the factors there are factors that are keeping it near the surface as well.
So the one below that's a time, they colored it based on time, right? In the blue image below. I
think the blue image, they color based on the type of movement. So like the yellow bits are if it's very close to the cell. So those are what they're classifying as skim events.
Gotcha. So red is going to be like when it's like really, really close and like lingering too much.
Okay. So I think like the idea is that there are like receptors there, but it kind of almost needs to drain away all that energy from the virus in order to get it to stop in one place.
The end result of this is it just leaves.
This is like an unsuccessful event.
That's amazing though, right? Like, so this is like crazy stuff going on in terms of like what they're able to see. It's very fast, right? This is diffusion. This is like a particle just like bouncing molecularly within the liquid.
And this is like where you see it skimming the surface, and it does go through periods of skimming and then freely diffusing and then skimming for a bit. And the thing that we're also interested in is it's not just about that area of it, but we also want to see how it does when it lands. And I think on the top bit is the timeline showing what the researchers think the behavior is. So it's what's skimming now. It's freely diffusing. It's going still above the cells, but it's bouncing up and down. But eventually, it's going to land, and that's like the interesting part.
Yeah.
Um,
One of the things that this paper kind of highlighted to me as well is that the surface characteristics of the cells probably have something to do with the likelihood that a virus is going to be able to make contact with its surface. I think they pointed out that like very rough cells sometimes increase the amount of dwell time that the virus might experience. Oh, like this.
yeah, like so this is like because instead of like bouncing off, it's bouncing within the crevices. So this is interesting because I know like immune cells tend to have quite kind of like really rough edges. You could explain that. I mean, there's lots of interesting like questions this like arises about like why certain things are like the way they are.
Yeah. I mean, it's like this is a unprecedented. That word is so common now. This is like an unprecedented view of the surface of a cell, and an actual like. I think that the greater field is called single particle tracking, right?
Right.
And so yeah, just like we've never been able to have these sort of high speed images of what's going on, and so that's going to let us really understand a bit better what are these first events and what factors go into these first attachment and entry events.
Yeah, I think here they're showing like membrane diffusion. So the idea is it's going through a membrane, perhaps, or between the junctions. I just find this fascinating to look at because again, yeah, this is like viruses in motion as we've never seen them before.
Yeah.
So
I guess I should say now that we've looked at a few, it's like so the motion of the camera is not the motion of the microscope for people, right? That's just like the digital, like to get a better view, it's like moving around the digital image so we can see all the angles. So the motion of the microscope is so that they can even get this dot, right? Like they have to use the stage so that they can always have like the viral particle in the right focal plane, and then also capture like the space around it. Why are the cells so static? Yeah, because I think it's about the cell. I think it's about the time scales here. That cells do move at some level, but not as fast as a viral particle can diffuse around.
Yeah.
And then the other thing is that they're staining for actin or something, right? Like that's the cytoskeletal elements. And so there are some parts of the cells that are like moving faster, like the membrane, let's say, like kind of it would be like wavy and stuff like that, but we can't see those waves in the membrane because what we're seeing is like a static image of like the cytoskeletal elements. And so actually in this, I think actually this is a good example of it. You can see there's like a donut that it formed. So that means that I think they have some better pictures of it in the paper itself, but they say that like they can actually see some of like this figure. They can see like some of those protrusions. I think they call them nano something. They can see protrusions of the cytoplasm by inferring that they're there because the viral particle is bouncing off of it. But there's no cytoskeleton. It doesn't actually get stained. So yeah, like there's definitely more motion than that happens in real life. Like there's a lot of motion going on. We're only seeing some of the motion because based on like how we have tags, how we have fluorescent tags to actually see certain things.
Yeah, because like when you fly, a fluorescent tag isn't like it doesn't light up the whole time. You have to continually give light to it, but it bleaches as well, which means that that there is like kind of like a refractory period in between like each of these things. So there is a time gap that we're not seeing.
So there's a time gap, and then there's also things that just aren't stained, right?
That's right.
Things that just aren't stained. And I think one of the fascinating things about this technology that they talked about was that they're getting around some of the photo bleaching effects. Like they're able to look at these things for like quite a long time, right? These are like several tens of seconds long videos. And actually, if anyone's spent time like imaging stuff in the stuff like that is the range where things bleach sometimes and you don't get to see everything. But the fact that they're doing these scans so quickly, like whatever their hardware setup is, they say that it's not enough dwell time with the laser in order to bleach things out. And actually they do have an issue when there's a long attachment period, then they can, if the virus doesn't move around too much, then they can bleach out that virus. because the
Right.
laser is just saying like continue training the laser on this one spot instead of like doing the scan, right? We saw in the first image they do like a sort of a tiny volumetric scan around where the virus could be. So I think that's like a really interesting. There's all these different like engineering trade-offs they were able to balance and bring together in order to give us this visualization, which isn't the entire story, but like is like a lot more than we've ever seen before in terms of like single particle tracking. Yeah.
Yeah, definitely. There's like this. I mean, again, I'm going to show another video,
yeah,
which is.
we could
I'm
have probably done the
gonna
whole
keep
hour
going.
of looking at these videos and talking about them.
Well, yeah, definitely. I mean, I think this is one of the sad things about the way scientific papers are presented, because there are these amazing videos for these things, but they're always like right at the end and hidden in the supplementary figures, so many people just don't get to see them.
Yeah, but
So,
again,
I now
if
consider
you're
that
interested in us diving deeper into this paper, then leave a comment in the description below or give us a toot. There's definitely a lot to look at in this paper.
And if you're an author who's had a paper with lots of great videos, but since the journals are published in paper, that they're just not being shown, point us to that paper and we'll show it for you. We'll sit down and just have a watch party of great, cool scientific videos.
I mean, you can also have a watch party with us, I guess. Now that we're on this aspect of asking how people should interact with us, like we'll host your watch party. You can come join us.
Yeah,
you can talk about what's going on.
We can just like talk. I mean, even if we only half know what's going on, which this is where they look at multiple virus movements in one image, which again, like I don't know how they did this, because as I understood it, like those virus motions is like the motion of the camera as it works to adjust to the field of vision. So I legitimately have no idea how they could do multiple viruses at the same time.
Yeah, I did read that they're able to somehow segregate. I read something about like part of it is in the data processing part, that when they notice that there's two, they're able to somehow focus on two pieces. Yeah, but I'm not 100% sure. Because the stage has to move, right? And when the viruses are so far apart, how does the stage move enough? Maybe the motions is so slow and the motor is so quick. But I'm not 100% sure.
Because they use a piezoelectric motor, which is like a rock that kind of vibrates essentially. So you can do very fine-tuned motions with it. So maybe it is just that their engineering is so fast that they can do look at multiple viruses at the same time.
Yeah, because I can imagine that, like, because this is something that we see in like the spinning disk style, or like a concept that I have from spinning disk is that like they are rastering right across the entire field of vision. But the big contribution that this paper does in the single particle tracking is that they raster in this very specific cloud around where they think the viral particle is. So and there's two cameras, right? One camera is doing the zoom out, and one microscope. One microscope is doing the zoom out, one microscope is doing the zoom in. So it's potentially that they raster between each of those points, right? Like they're just like boop, 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 and then they go over to where they know the other one is. Okay, I think we should move on, because we could probably look at these forever. But
yeah,
we have like
I think
a couple more papers, and
we've got a couple more papers to get through. Let me quickly pull up the.
So the next one we have is also another kind of interesting view that we don't normally get to see. Antibiotic polymyxin arranges lipopolysaccharide into crystalline structures to solidify the bacterial membrane. So here we're talking about antibiotic, polymyxin, also known as, or polymyxin E is also known as colistin, which is sort of one of these last line gram-negative antibiotics. Polymyxin B is also very common. I think I've seen polymyxin B, and it's actually also used in like a bunch of studies about membranes because it's a useful interaction with them. And so what they do is they get some of these they're actually using outer membrane vesicles here from a gram-negative. So it's not the bacteria themselves, but rather blebs of the bacterial outer membrane. And they set that down onto a very smooth surface, a piece of mica. And then they use atomic force microscopy to scan over that. So that's like a tiny little record needle that goes over everything. And if it gets close, then it gets pushed up because of electrical interactions. And they're able to see like basically how much does something stand up from the surface of the very, very smooth mica. And when they add polymyxin E, and they look at these outer membrane things, they create this weird ass honeycomb stuff's going on. So essentially, parts of the membrane are being forced up into these ridges, and that is very curious because we know some stuff about how polymyxins work, but we don't know the whole story. And this is really quite a detailed view to be able to figure out what's going on.
Yeah, so essentially they find that the polymyxins are somehow entering into the polysaccharide layer and then interfering and like changing the structure, causing like almost polysaccharides to crystallize in these hexagonal kind of structures, which kind of messes up firstly it pokes them out of the cell wall, and so it makes the bacterial membrane all bumpy and weird, and makes it less able for the bacteria to function essentially, and makes it more likely for there to be for it to pop essentially.
Yeah, they do a bunch of interventions, like the one that we were just looking at was they made mutations to the bacterial part, and so that they can try to see like what part of the LPS is essential for these weird structures to form. And then in the next figure, they change the polymyxin B and like give it all these different modifications, make it shorter, change the charges on it. And they also see like, some things are important and some things aren't important in terms of like making these weird patterns appear.
Yeah. And I think this is like really interesting, like, and how it kind of interferes on a molecular level. And they do a lot of confirmation studies to see whether this actually is killing the bacteria or there's just like awkwardly inconveniencing the bacteria, and there's some other effect that's really going on.
And it does seem to be that the honeycomb structures they're at least sufficient for killing. Yeah, because they have like one example that stands out that is like, oh, it didn't really go. Let's see. I think
interesting because they had because I think the size of the structures matter and like how they're oriented. So one of the mutations that they made, I think it managed to create the hexagonal structure without killing the bacteria at the concentration you expect.
Yeah, so there's definitely more. There's probably a little bit more to the story than hexagons hexagons make the membrane stiff and kill these bacteria. But that is more. This is more confirmation of this than we've ever had before, of this aspect. The stiffening of the membrane.
And it'll help for designing new antibiotics, because once we know that this is what's doing once we can model what this is actually doing to the membrane, then we can better understand how to design other antibiotics to hit in maybe a different way to create crystalline structures in the membrane.
Yeah, totally. Like I think it's reminding me of the optical tweezers paper that we looked at when they were like looking at how do these like inhibitors inhibit polymerases. In some ways, this is like a totally different set of tools and a totally different physiologic system as well. We're looking at membrane dynamics, but the same sort of thought process can be applied, that like if we have these specific tools to look at a specific physiologic process that we want to target, then we can start saying like, well, what if we use like a bunch of other compounds in here? Like, could we replicate, right, this sort of structure in a different way? And does that give us a new class of antibiotics in some way? Yeah, something like that.
Yeah, I mean, I can imagine someone right now going, okay, let's put all this structure data, all our chemicals on our back catalog. Like, imagine like this pharma company exec with like a cigar, being like, we got a bunch of chemicals. There's nothing in there. Right, let's go ahead and using AI to find whatever chemicals are similar to these, and then we'll hit a bunch of OMVs with them, and then we'll see which ones create weird structures, and then we'll sell them. We'll sell them to people.
Yeah, exactly. That's happening.
If it's not happening, then I'm going to do it, and I'm going to make billions.
I will say though that I got really excited, but then I forgot that one of the good things about the optical tweezers that we looked at was it was in a flow cell, which meant that you can do like more high throughput stuff. And this is actually they had to gently set down bubbles of bacterial membranes on a very flat surface and slowly scan over with a little stylus. So maybe that doesn't scale up in the same way.
Yeah, probably doesn't scale up. Damn it.
I got excited. Okay.
Never get excited in science.
Let's look at the next one. We got a really short, almost just like an observation, a type of like data observation. Phage tRNAs evade tRNA-targeting host defenses through anticodon loop mutations.
Yeah, this is a preprint, so it hasn't been peer-reviewed yet, but it's just like it came up with a really interesting theory because there's this observation that phages encode tRNAs. So tRNAs are an important part of forming proteins. They kind of translate nucleic acid code into amino acids. And the idea is that, well, people didn't really know why phages have these tRNAs with them, because surely if they infect a host should have enough like tRNAs around to make proteins. Because otherwise the host wouldn't be able to exist. So why would phages need to make these on their own? So one theory was that codon optimization. So there's this idea that different living things need different abundances of certain amino acids. So in order to like balance that out, if you want to have a cell create a specific protein you want to make sure it's got the right number of tRNAs there, so when a protein is being encoded, there isn't a hold up to the process because they're waiting for a specific tRNA that isn't created by the cell in order to translate the whole thing. But this paper has kind of looks at it slightly differently because they notice that certain host cells create phage tRNA kind of dissolving enzymes, so tRNAs, I think they're called, or something like that. No, they're not called that. They're called something different. But the idea is that enzymes that specifically chew up RNAs, and the idea is that so that when a cell gets infected by a virus, it wants to commit suicide so that it doesn't infect any other cells. It wants to draw everything to a stop, so that includes its tRNAs.
Yeah,
and so
tRNA anticodon nucleases.
Yeah, that's right. tRNA anticodon nucleases. So the idea is like these will dissolve the host's own tRNA nucleases so that it doesn't allow the virus to take advantage of its systems. So they theorize that phages must create their own tRNAs to compensate for this. But they thought, following on to that thought, well, these tRNAs would have to be resistant to those codon tRNA nucleases in order for them to survive. So they did some sequence gazing to see whether the phage tRNAs had the active sites that would allow them to be broken down by the nucleases of the host. And what they found was that they didn't. That a lot of them were resistant to anti-nuclease activity. At least they looked only at microbacteria, and their phages, and so that's what gave them this idea to like publish this preprint, which sets out that theory, and they're still kind of get the rest of the data.
I mean, I think I'm a big fan of publishing in this style. It's like once you find something interesting, get it out there, start the discussion, let other people know. Like, you're doing the work, and like you wanna keep the conversation updated. You might get feedback for this type of work from your peers that helps you grow your particular thing. New collaborators, right, could like come from publishing this stuff. Anyways, I just want to say that like I really appreciate when folks do this type of publication because yes, there is competition, but it doesn't have to be all competition, right? There's a lot of like collaborative opportunities that get missed if you're just hoarding your data for like that one big tome that you're gonna publish, right? Like at the end of the year or something like that.
So which brings us to our next paper. The discovery of integrons in Archaea, platforms for cross-domain gene transfer.
Yes. I mean, in some ways, this is also a little bit of sequence gazing, where they looked at a whole bunch of archaeal, I mean, these days it's not genomes, it's these metagenome assembled genomes. So, there's a few caveats there, but they try to do a lot of data cleaning to make sure they're not getting some false positives sort of deal. So they look across all these different archaeal metagenome genomes, assembled genomes, and they find the signature of these integrons. So integrons, they're known in bacteria. They have a very specific structure where they have like an integrase, an attachment site, and an insertion cassette, and then they can flank a whole bunch of other genes. So it's a mobile genetic element that is essentially thought in bacteria. It's known that it like it moves cassettes around, especially antimicrobial resistance and stuff like that. And so they're also in Archaea, is what they find in their sequence gazing. And I think the most interesting part of this, or like what I love about the paper, is that then they show it as well that it's possible for the archaeal sequences that they discover to work in E. coli. So Faz was just showing us that that figure where they. Go ahead.
Oh no, sorry. Is this the right figure? Figure 5, the one where
Oh
they
no,
got this.
no. Figure two.
Figure two. Okay, I'll go all the way back.
Phages was showing us the figure 2, where they put it inside of bacteria, and they're like, whoa, these archaeal sequences can move stuff between bacteria. So that really opens up this idea that bacteria and archaea are having a set of genes that are jumping between them. Kind of interesting from the phylogeny point of view. The integrase that they use is its own individual, is a monophyletic group. So it's like they have their own integrase, but they have a lot of mixing with the attachment sites and the ATTC. So like the stuff that they get excised off of and the things that they insert into, those locations on the genome are kind of mixed up between the bacteria and the archaea, but the bacteria and archaea each have their own proteins that do that work of cutting things out and putting it in. So yeah, I mean, it is, I don't want to linger too much on this, but it is a really interesting piece of news in the sense that there's this identified potential for gene flow between archaea and bacteria. And there's lots of genes that have been identified in these integron structures that people don't know what the function is. Like there's more genes, people know more about the general genome than stuff that's inside of this particular range of things. And they could be really important, right? Like these are the genes that are getting swapped between organisms, probably for adaptation. I mean, well, again, that's me thinking everything as a function, but hopefully for adaptation, or likely in an adaptive capacity, they have some interesting functions. What are they? Why don't we know more about them? And what's going on? So I think it's a really interesting perspective. Figure 5, if you wanted to go back to that one, that's just saying that of the things that they are able to annotate, it's weird, but they cluster by the environments that these samples were found, right? These are metagenome assembled genomes, right? So they came from some environments, right? The sequences have a provenance to certain environments. And so they're just seeing that, the functional annotations they are able to extract from this cluster with those environments. So maybe it means that, right, again, that's kind of like going down this thought. It could be adaptive in these environments in some ways, right? This might be a clue to help us identify what these proteins even do, because we know that we find them in these environments, right? And they're being swapped. So yeah, that's this. It's actually a really big finding, I think. And I'm sorry, like I can't like dwell upon it any more than we are right now. But maybe if you're interested again, just let us know and we'll try to talk more about it.
Because Archaea are kind of our ancestors, so the thought arises of, okay, so could integrons work in us? Like that's the kind of
yeah. Or I think that's also a really cool, like again, going back in that deep time and the origins of life. Yeah, like Archaea sit on the phyletic branch between us and bacteria. Yeah. Right. So it is interesting to think about that as well. Okay. And our last little paper for today is also just a little update, a news update, a horrifying news update, where a Shigella sonnei clone with extensive drug resistance associated with waterborne outbreaks in China. So there have been several outbreaks of Shigella. Dysentery is the classic disease here, between Shigellosis, between 2015 and 2020. So in this paper, we actually got referenced this paper through Mastodon. So we want to bring it up because maybe people in that group were interested in talking about it. Essentially, they do sequencing, very deep, full genome sequencing of all these different Shigella strains throughout time and throughout outbreaks. And they're able to look at some patterns that emerge. One pattern that they say is they're all from one clade. This is China-specific outbreak Shigella. Which I guess really points to the scope of the problem that's occurring here, that like it's a public health concern, right? That these things are appearing and they're all geographically located. All of the patient strains also have inside of the clades, if we go down to figure 2, there's like this Christmas bar, the green and the red bar that goes, it's like the 4th bar in, the green and the red. So all the green ones are patient samples, and all the red ones are from water samples. So in every major outbreak clade, there's a water sample that's closely associated with it. I mean, people knew that it came from the water, but it's like, this is like a more evidence just being like, yeah, it's untreated water that's likely spreading these particular outbreaks.
Yeah. Very much modern Jon Snell kind of paper, I think.
Yes.
They are like, there is a bit of detective work here where they're going around looking at each of these outbreaks where people have like gotten hurt, and they try and track down. And I think there's quite a lot of things that are related to water, and also there's like a lesson about antibiotic stewardship there. They're concerned about.
Yeah. Well, if you just linger back on figure 3 that you scrolled through there, then they also go in and they look at the antibiotic resistance on these particular bacteria. And like, they also assort by clade. Interestingly, I pointed out, we were talking about colistin recently. The colistins is like last round antibiotic that's often used for gram-negatives. But there are resistance cassettes that are floating around now. And so actually a bunch of these Shigella have that resistance cassette as well. That's not good news. The authors actually have a nice discussion. They briefly touch on the fact that China uses colistin in their agriculture, but they recently stopped. They're not going to use it in their agriculture anymore, but they are going to start using it in humans. Well, that's part of the trade-off, is they say, no more agriculture, now we want to put it back in humans. But of course, that dynamic, who knows if that dynamic's going to shake stuff up. The authors suppose that it might increase the amount of colistin resistance that goes around, just because I guess it's being used closer to the selective environment where we would expect human pathogens to be. But yeah, lots of interesting stuff. Very important, I think, for public health concerns, especially if public health in China, right? But also lessons to be taken to other public health systems in terms of outbreak strains and their relatedness. It's just quite a nice and large data set of Shigella.
Yeah, like the importance of like how water treatment and because like, even as we get more advanced, new things can crop up. That means that we need to think about how we supply like utilities to everybody without causing outbreaks like this.
Totally. And I mean, I don't think we need high-tech techniques to tell us that, but certainly more evidence doesn't hurt to be able to tell us that, clean water is very important. And also, I think as outbreaks occur, this is also a really useful data set to help public health officials there, like track the source of things, right? Like we saw that during the COVID epidemic, or we are seeing that during the COVID epidemic, that genomic surveillance is helping us say like, oh, we think that it's coming from these localities, maybe helping set some policy. I mean, China has a lot more control over the migration in their country. And so maybe they'll be able to use this data to some effect using that particular tool. Again, value judgment aside, like whether that's good or not, I'm just thinking about the ways in which this data could be used.
Yeah, I think that there are a lot of like interesting societal things to look at, but also like just looking at the science of like how these plasmids seem to accumulate in outbreaks and
how they're evolving into their own distinct sets, and just I guess the other concern that the authors bring up is the resistance that they identify. It is moving on plasmids, and we know that plasmids in the Enterobacter move between Enterobacter. So like Shigella is classic for giving stuff to E. coli and back and forth, right? Also Enterococcus, Klebsiella, these are things that, already big problems. Gram-negatives and their antibiotic resistance plasmids, just a huge thing. I'm going to put a paper actually about that on the docket for next week. So and that's all I want to say about that. So join us in 2 weeks where we're going to just cover much more cool microbiology news.
Yeah, exactly. We want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know.
Yeah, I totally agree. You can reach out to us on Twitter or Mastodon. Hopefully Mastodon these days, but we're no judgment there. We both believe that peer review is a process and that it requires folks to have read the papers to make it work. So we want to read with you guys. And so if you think you have something to add or found something unclear, just let us know.
It's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Tune in 2 weeks for more microbiology content. Until then, goodbye.
Bye-bye.